You take mitosis or meiosis, it doesn't matter. There is a bit difference. Okay, that is the number of chromosomes present inside a cell after the end of mitosis or meiosis. Okay, in the end of the mitosis, what you see is the same number of chromosomes will be present in the daughter cells. But after the end of the meiosis, you see half the number of chromosomes present inside the daughter cells. That is the difference. Okay, now let us imagine this is a normal cell. Okay, somatic cell, easy to understand. And the nucleus containing a thread-like structure, what you call? You call it as chromatin. So, what is exactly this chromatin? Before we understand the structure of the chromatin, what exactly a picture comes to your brain when I talk about the nucleus? Very simple, X-shaped structures, right? That is nothing but called as chromosomes. Okay, but when exactly you see the chromosomes? Do that X-shaped structure present inside the nucleus all the time? No. It is not present all the time inside a nucleus. When the cell is undergoing metaphase stage, only in the metaphase stage of the cell division, you see X-shaped structures. Okay, let me just show that to you. This is now called as chromosome. Yes, it looks like X, right? Okay, so this is chromosome. I have got 4 chromosomes here. Got it? Okay, so many chromosomes, X-shaped structures. Okay, I'll put it here. I cannot stick it, but I'll show you. Okay, so this is now X-shaped structure seen only in metaphase. It is seen in mitotic metaphase, at the same time meiotic metaphase also. Okay, how many chromosomes you see inside the nucleus in the metaphase stage? 46. You see 46. Okay, see the parts of the chromosome. This is actually called as the centromere. Centromere actually holds the chromatids. Okay, now what is exactly chromatid? So this is actually a chromatid. Okay, this is one chromatid and this is the other chromatid. I can easily separate them. Very simple, let me tear it. So I'll tear this. Now X-shaped chromosome has become linear chromosome. See, this is now one chromatid. This is another chromatid. So, when in a chromosome you see 2 sister chromatid. Why sister chromatid? Simple. Whatever the genetic information it has got, that is nothing but in the form of sequences. Adenine, guanine, cytosine, thymine, adenine, guanine, cytosine, thymine, adenine, like that. The sequence goes, I'll tell you, I'll show you. That is present in this chromatid, sister chromatid. The same sequence, whatever the sequence present here, exactly in order, that should be present in this also. So they make up the sister chromatid. Got it? So finally I can combine it. So with the help of the centromere, now centromere holds these 2 sister chromatids, makes a perfect X-shaped chromosome. Got it? Understood? But now what is there inside this chromosome or chromatid? Hope you understood. This is one chromatid, this is the other chromatid. Both are called as sister chromatids if it is present in a single chromosome. Now the interesting thing is, let us imagine this is chromosome number 1 and this is chromosome number 22, some other chromosome. Okay, so normally size also varies. So to make you understand, let me just tear some part because the size matters here. Normally chromosome number 1 will be largest chromosome, whereas the chromosome number 21 will be smallest chromosome. Okay, so the size decreases as we go from chromosome number 1 contains 2 chromosomes and 22 also contains 2 chromosomes. Always chromosome occurs in pairs. Got it? If I say chromosome number 1, you have to imagine 2 chromosomes. Same chromosome. Got it? But 2 copies. 22 also got 2 copies of this chromosome. Copies of this. Okay. See the size. Varies, right? Now, interestingly, let me just tear this. So I have separated the sister chromatid of the 22nd chromosome. So this is sister chromatid of 22nd chromosome. This is the another sister chromatid of the 22nd chromosome. So they are combined together by centromere, forming X-shaped chromosome. But these 2 chromosomes are entirely different. Now I have taken a sister chromatid from chromosome number 1 and I have taken another sister chromatid of the chromosome number 22. So let me just bring it now very close to each other. But they will not combine. Why? The thing is, this becomes non-sister chromatid. It is not sister chromatid for this chromosome or chromatid. Both are entirely different. See the size. It's matching, it is not matching. Yes, so it's not possible. So two chromatids only combine when their genetic sequences, that is the sequences I said, should match. Okay, making a perfect X-shaped chromosome. This is seen only in metaphase stage. Then what kind of structure you see them in other stages? Particularly cell cycle starts with interphase, then enters into the M phase, that is prophase, metaphase, anaphase, telophase. Okay, here to understand that, let me go with its simpler form. Okay, now we will imagine this is the chromosome containing two sister chromatids. Okay, I have taken a tiny patch of this chromosome, tiny, very tiny. So we'll tear it. Okay, so it's a tiny piece of this chromosome I have picked from a single sister chromatid. See, a piece is removed. Can you see it? Okay. Now, what you see inside this tiny piece? It's gone. Okay. You see this. What is this? This is the DNA. That means the DNA inside this chromatid will be coiled in such a way they are compressed and the length will be huge and finally makes X-shaped structure. Got it? Okay. So here let me just make it linear so that it will be easier for us to understand. This DNA mainly contains two strands. This is in another one strand of the DNA. This is the other strand of the DNA. Okay, it contains certain sequences. You could see some alphabets here. A is there, A, T, T, C, A, T. What is this? You have already studied in your previous year. They are called as nitrogenous bases. In DNA, you see four nitrogenous bases: adenine, guanine, cytosine, thymine. Adenine can be written as capital letter A, thymine capital letter T, guanine G, then cytosine C. These four will be there. Okay, but look at the arrangement. It is not like adenine, guanine, cytosine, thymine. It will not go in order. See this: adenine, guanine, thymine, cytosine. Cytosine, guanine, adenine, thymine. Cytosine, adenine, cytosine, thymine, adenine, guanine, thymine, adenine, cytosine, guanine. See, it goes like that. Yes. So this is in one strand. The sequence is present in one strand. What about the other strand? I hope that it is visible to you. The other strand contains its complementary base pair. Adenine always binds to thymine. Got it? They are complementary. You will understand it in higher classes. The guanine always binds to cytosine. It is complementary. Or cytosine always binds to guanine. Cytosine cannot bind to adenine. Or adenine cannot bind to cytosine. It is rule. Thymine always should bind to adenine, or adenine always binds to thymine. Guanine, cytosine, or cytosine, guanine, bonding is possible. Got it? So this is the thing. So you could see different types of sequences and their complementary bases. Got it? And these two sequences, nitrogenous bases present in different strands, are joined by hydrogen bonds, so that the DNA looks like a ladder. Yes? Ladder, right? Okay. So this is the basic to understand. Okay, now the question is, what is the charge of the DNA? Need a chalk, please. The charge of the DNA is always negative. Very important, please write it. Charge of DNA is always negative. Why DNA is always negative? I think not necessary, but let me just give you the idea. The DNA mainly contains phosphoric acid group which is always negatively charged. Hence the DNA is negatively charged. Okay, not now. In the second PUC you will understand it. Okay, let me not confuse you. This is the simple basic model of the DNA. The DNA looks like this. Got it? So I have picked this DNA from a tiny patch of the chromosome from the sister chromatid. Yes. Okay. We'll fold this. Now, how exactly the DNA is arranged? The DNA strands are arranged like this. That is double helix model. Yes, see this? They are coiled spirally, spherically. See the coiling. Wonderful, right? So this is the thing. So looks like spirally coiled staircases. Yes. Okay, it is spirally coiled. The DNA strands are spirally coiled. It is because of the arrangement of the nitrogenous bases and other groups. You will understand it in second episode. Don't worry. So we are going to the next level, correct? See here I have used color. So red color is adenine. This black color is thymine. Thymine, adenine. Adenine, thymine. This is cytosine. This is guanine. Guanine, cytosine. Cytosine, guanine. See the arrangement? You can take the screenshot if it is visible to you. See, this is actually the thing. The length goes like that. As I said that inside our cell, if you take the nucleus containing a huge stretch of the chromatin, that is nothing but our DNA itself. What is the length of the DNA present inside our each cell of the nucleus? 2 meters. What is your height? Hardly 5.3 to 6. Just imagine. Huge for a cell. Okay. Next. Now the question is, just let us imagine this is now chromatin containing DNA. Okay. The next level. So this is the cell. If I ask you to put this inside the nucleus, is it possible for you? No way. For this you need to compress this, right? We need to compress this in such a way. Okay, I need to fold it. If you just uncoil this, obviously the length will be around 2 meters. Okay, I will fold this. This is nothing but called as condensation. Have you heard it in Profess? Condensation of the chromatin occurs. This is condensation. Got it? Okay. So because of this condensation, I can easily fit this chromatin, 2 meters chromatin inside the nucleus. So who helps in the condensation of the DNA? This is very important. Okay. So now we go to the next level. For this, I need this. See. Please do understand this. This is actually the actual structure of the DNA. Okay, that means I have got a huge thread here which contains 2 threads which are spirally coiled, helically coiled. Okay, so helically coiled, and the length is very long, approximately around 1.5 meters. Okay, so this is the real DNA. So I have taken this DNA from a small patch, from a segment of this entire length. Got it? This is magnified image of this. So now you know this is now DNA. Correct? This is now DNA. The length is very long and it's hard to put them inside the nucleus. As I said, it has to be compressed, condensed. For that we need certain help. What is that? So we need the help of certain proteins. They are called as histones. We need the help of histones. Here I have a tennis ball here and I have marked this. I have put around 8 circles. 1, 2, 3, 4, 5, 6, 7, 8. 8 are there total. Each circle is nothing but a type of protein. This is one ball. Okay, one ball. And it has got certain circles inside. This is the one particular part of the ball, the visible part. This is the back side of it. Okay, so let me write the back side of the ball, which also contains 4 tiny circles. These circles are nothing but called as proteins. P. So how many proteins are there in one ball? 1, 2, 3, 4, 5, 6, 7, 8. So this ball contains 8 proteins, and these 8 proteins are arranged in such a way finally makes a big ball. Got it? This big ball is called as histone. Got it? That means in histone there are how many proteins? 8 tiny proteins, which makes up a big histone. This is histone protein. Okay, the name is H2A. Here, let me write H2B. Let me write H3, H4. One side of the ball contains H2A, H2B, H4, H3, H4. One side. Okay, and here let me write the same H2A, H2B, H3, H4. These tiny histones, how many are there? 8 are there. So we call it as histone octamer. So finally makes a big ball, which is called as histone octamers. Got it? Understood? So this ball is now histone octamer containing 8 tiny histone proteins. What are they? H2A, H2B, H3, H4. Done. And I am holding a marker pen here. This marker pen also plays an important role. For example, let me write, this is now histone octamer. Let me write HO, histone octamer. Here, after some interval, you see one more histone octamer. Again, you see one more histone octamer. Again, one more histone octamer. and you will see millions of histone octamers inside our nucleus. Millions. Got it? Okay. Just before histone octamer, you see a linear protein like this. Looks like 10 now. You just imagine 10. See, one more protein here, and one more protein here, and one more protein here. See, looks like 10. So what do you call for this? This is histone octamer containing 8 histone proteins. And this is the linear histone protein. Another histone protein is there, but it is not attached to this histone octamer. It is separate. So this is called as H1 protein. Now the question is, how many histone proteins are there? How many histone proteins are there? Let me write HP. Not Hindustan Petroleum. It is histone proteins. We have 5. H1, H2A, H2B, H3, H4. But here you see the copies. 1 copy, 2, 3, 4. Total making it as 8 histone octamer. And this is only one. That is always present just before the histone octamer. So H1, here also you see H1, H1, H1. So this is marker pen is H1. This is histone octamer. Got it? Okay. You see this pen-like structure. That is H1 and histone octamer many times, millions of times. Okay, inside our nucleus. Now, I said the DNA is very, very long. It will not fit inside the nucleus. So we need to condense it, compress it. That means I need to reduce the size, but without damaging the DNA sequences. I should not damage this. I cannot take a scissor and cut. Okay. So here histone octamer plays a very important role. First thing is, so this is the starting point of the DNA. Let us just imagine. And first thing is, okay, rolling is a bit difficult. It first coils around this H1. See, 1. It goes, wraps around this H1 and completes one turn. See, one turn is completed, correct? So one turn is now complete. It wraps around H1, that is one turn is complete. DNA completes its one turn around the H1 protein. Next is it wraps around the histone octamer. Second, see, one turn is complete, almost complete. Okay, now it is holding is a bit difficult. It may fall. One turn is now complete. Now it goes for the second turn, but it will not complete the complete second turn. Remember it? It wraps around the histone octamer only 1 3/4 times. Not complete 2 complete rounds. But around the H1, DNA complete its complete one round. Got it? Okay. So finally it comes out. It comes out like this. And finally it meets one more H1. Again it wraps around it for one time and then it wraps around the H1 octamer. Like that. It goes like that. So this is the thing you need to understand. Got it? Now this structure, when the DNA wraps around H1 and histone octamer, what you call for this only structure? Okay, so this is the protein, this is also protein. So DNA wraps around this histone H1 first and then histone octamer. Then this structure is called as nucleosome. Okay, so this is the DNA. DNA now wraps around this H1 one time. See, one time. Please note this point for future, very important. Then actually it wraps around the histone octamer. How many times? One time is completed. Next, it will not complete its full turn. Okay, it escapes. That means 1 3/4 turn. Okay, around the histone octamer and goes and meets another H1. Again the same. Again 1 3/4 times. Again. Okay, now I am talking about this structure. What do you call for this structure? Which contains negatively charged DNA. DNA is now negatively charged because of the presence of phosphoric acid, which is negatively charged. And this one, this histone octamer, which mainly contains two important amino acids. We know that the basics. All proteins are mainly made up of amino acids. Yes. So these histone octamer, that is, you take H2A, H2B, H3, H4, doesn't matter actually. Okay. All these are mainly contains two important amino acids which gives them positive charge. So histone octamer is positively charged, but DNA is negatively charged. Why? Because the amino acid present in the any type of histones present in the histone octamer are lysine and arginine. If you want, write down. Lysine plus arginine gives positive charge to histone. So entire histone octamer is now positively charged. So negatively charged DNA wraps around the positively charged histone proteins, that is H1 plus histone octamer, then this structure is called as nucleosome. Please note this one. We call it as nucleosome. Done. How many nucleosomes are there? 2, 3, 4. In this diagram, there are 4 nucleosomes. Finally, many nucleosomes are finally makes this DNA highly condensed. Now, the DNA looks like this. It looks like beads on string. Beads on string. String is now compared to DNA. Beads are compared to the histone. Okay, it looks like syrup. Okay, so simple. It looks like this. Histone octamer. Finally, DNA and histone octamers are called as nucleosomes. So here we have got so many nucleosomes, correct? All these nucleosomes helps to condense or compress the DNA, and this structure is now called as solenoid. What do you call? Solenoid. Oh, I don't have dust. Please write down. Many nucleosomes finally makes up highly condensed DNA called as solenoids. Got it? This one structure is called as nucleosome. Many nucleosomes are called as solenoids. Got it? Now let me just erase this. Hope you understood this. Basics, very important. Otherwise you cannot understand cell cycle and cell division. Basics are very important. Catch this. Take your time. Be patient and watch. Okay. We'll keep this aside. Not required. Okay, now the diagram. Let me just draw the diagram. What is this now? Solenoid. Highly condensed DNA. The DNA structure is becoming very, very complex now. It's very difficult to understand the structure of the DNA here. Yes. But again, the length is more. Again, I need to compress it. That is for the third time. I am compressing the length of the DNA. Third time. First time is nucleosome. Second time is solenoid. Third time is I need higher level proteins. They are called as non-histone proteins. That means I need a type of protein which are not histones. I need non-histone proteins. Some examples for non-histone proteins are scaffold proteins. What is this scaffold protein? Okay, so very simple. This is now scaffold protein. Can you see this? Okay, looks like a spring. Okay, they are also spherically coiled, but they are not double helix, single. Only one strand is there. This protein is a long linear protein, but they are spirally coiled like this. Looks like a spring. Can you see this? Okay, looks like a spring. Let me draw again. The scaffold proteins will be like this. Sorry. Again, I made a mistake here. Keep this. Like this, like a spring. This is scaffold protein. Now, this solenoid structure now goes like this in such a way that it goes parallelly with the scaffold proteins like this. See, wonderful, right? So it is the third level of compression, compressing the DNA. See, what is the fourth level again? We need fourth type of proteins actually. Again, they are non-histone proteins. It could be DNA polymerase, for example. For the fourth type, we need a type of enzyme called as DNA polymerase enzyme. You will not understand what is this DNA polymerase enzyme. Okay, this DNA polymerase enzyme again compress this. So they actually pull this scaffold protein, these two scaffold proteins very close. So as a result, the distance is now reduced. It looks like this. Again condensed. Okay, the DNA is present here. Okay, so that is because of the DNA polymerase. And ultimately what you see is, you see a structure like this. When the cell undergoes mitosis, you see a structure like this. What What is the structure? It looks like X-shaped structure. So this is chromosome. Done? Got it? So this is all about the chromosome structure. No doubts, nothing else. I am not going to explain this again once the college reopens also. So I hope that this entire concept is clear to you now. Got it? Okay, so this is now chromosome. What do you call this structure now? This particular arm of the chromosome, you call this as chromatin. This is chromatin. Okay, then what is chromatin? Very simple, this is chromatin. Got it? Chromatin contains DNA, histone proteins, that is histone octamer everything, then non-histone proteins. Everything makes up a structure which is called as chromatin. So this is chromatin. Okay, and one arm of the chromatin is called as chromatin. Chromatin. This is chromatin, this is another chromatin. Both are matching in their nitrogenous base sequences. So these are now held together by an important structure called as centromere. Centromere, and now it become a chromosome. That's it, easy. So two sister chromatids, they match in their nitrogenous bases. Hence they are attached by centromere, finally making a X-shaped structure called as chromosome. And this structure is seen only in metaphase. Then what do you see in other phases? If you take prophase, this is prophase. Okay, nucleus, and the nucleus is slowly disappearing. Fine. So you see this. Only chromatin you see, no chromosomes. Okay. The question is, then if see this, it's nothing but like a small string. Okay, so we will put this out. So let us imagine, not DNA. This is now chromatin. Okay, chromatin. Everything is there now. Proteins are there, chromatin. But how they now separate to make 46 chromosomes? This is bit something is there actually. Simple. Let us imagine that these chromatins, they actually coil and make a structure like this. Looks like X-shaped structure. Once they form X-shaped structure, they cut, they separate. So again, one more X-shaped structure. That means they separate. Got it? Again, when they enter into prophase, again they combine, making a long chain like this. Okay, in the metaphase, they all separate. That means actually, if I pull this somewhere else, there is a chance that this chromatin can break. Somewhere else, the DNA is weak, so that it can be helpful in the making of the X-shaped structures, that is chromosome. At the same time, somewhere else, at particular point, looks like a gum, so that after metaphase or maybe anaphase, telophase, they rejoin, making a long chain like this. This is the thing happening. Got it? Hope you understood, and I think the entire concept is clear to you. Thank you very much.